Hello, everyone. Thank you for joining today's session. Our topic is ICH Q5B, analysis of the expression construct in cells for rDNA-derived protein products. This guideline provides a harmonized framework for ensuring the quality, safety, and consistency of recombinant DNA-derived protein products. Today, we'll break down the essential requirements and practical implications for professionals like us in the biotech and pharma sectors. The Q5B Guideline, adopted in 1995, is part of the International Conference on Harmonization's Quality Series. Its purpose is very focused. It outlines expectations for analyzing expression constructs used in the production of recombinant DNA proteins. This is not a complete quality standard for all aspects of biologics manufacturing. Instead, it zooms in on construct analysis, which is critical for assuring product integrity. To set the stage, let's define a few key terms. The expression construct is the vector containing the coding sequence and regulatory elements necessary for protein expression. The master cell bank, or MCB, is the original pool of production cells. The working cell bank, or WCB, is derived from the MCB and used in routine manufacturing. Finally, in vitro cell age measures the lifespan of production cells from thaw to harvest. Understanding these terms is essential for following the guideline's requirements. So, why is this analysis so important? Expression constructs must maintain the correct coding sequence throughout cell culture. Mutations can occur, and even minor changes in protein sequence may lead to serious safety issues for patients. That's why both nucleic acid and protein-level analyses are required. Together, they provide a full picture of sequence accuracy, structural integrity, and product consistency. The guideline highlights several analytical tools. DNA sequencing to confirm coding regions and insertion sites. mRNA, or cDNA analysis when multiple construct copies are present. Restriction mapping and PCR for copy number and structural integrity. And protein sequencing to validate the final amino acid sequence. The combination of these techniques provides confidence that the product matches its intended design. The guideline requires manufacturers to fully describe the construct and cell system. This includes the origin of the DNA sequence, the steps used in construct assembly, and a complete plasmid map with functional elements like promoters and enhancers. It also requires detailed documentation of how the construct was introduced into host cells and the criteria used to select production clones. Cell banking is central to biologics manufacturing. The MCB serves as the foundation, while the WCB ensures consistent production under controlled conditions. Both must be thoroughly characterized for genetic and phenotypic markers. Detailed histories, storage conditions, and culture methods must also be recorded. This ensures reproducibility and compliance with regulatory expectations. Once banks are established, genetic stability must be confirmed. This involves checking for insertions, deletions, copy number variations, and verifying the integrity of the coding sequence. Depending on the system, testing may involve direct sequencing or recloning and analysis. The core expectation is clear. The construct in the banks must match the original design within the detection limits of current methods. The guideline also requires defining the production lifespan of cells. This is based on data from pilot or full-scale studies. Beyond a certain age, cells may accumulate genetic changes, risking product variability. Verification of product integrity at defined cell ages, through either nucleic acid analysis or final protein analysis, is critical before extending production limits. In summary, Q5B stresses the dual importance of analyzing both the construct and the final protein. This two-pronged approach ensures product quality and protects patients. For manufacturers, compliance with Q5B is not just regulatory. It's a cornerstone of building trust in biologic therapies. To make things easier for teams, the guideline also defines a glossary of terms like expression construct, flanking control regions, integration site, and pilot plant scale. Having a shared vocabulary across teams ensures consistency and clarity in regulatory submissions. You can download the guideline reference document from link in description. Let's end with practical implications. Always maintain robust regulatory documentation. Validate analytical methods thoroughly. Ensure complete traceability across cell banks. And integrate genetic stability monitoring as part of ongoing quality assurance. By following these principles, organizations not only meet ICH standards, but also position themselves for sustainable, high-quality biologics production. Thank you for your attention. I hope this walkthrough of the ICH Q5B Guideline gives you a clear, practical framework for construct analysis in biotech production. Now, I'd be happy to take your questions in comment box.